Good morning, everyone. I'm Alex Capron, the chair of the Primer Board, and I have the pleasure of serving as the moderator for this special additional session on tough questions about Ebola, scientific, regulatory, and ethical issues with experimental treatments and vaccines. All of you who heard yesterday morning's keynote by Tony Fauci got some introduction to this, and many of you have read of the debates in the literature. We are not having a debate, but we are going to have people taking different views and trying to bring in all three of those considerations, scientific, regulatory, and ethical, in looking at the issues. Our panelists are in alphabetical order: Edward Cox from the Food and Drug Administration, Walter Strauss from Merck & Company and the immediate past chair of the Primer Board, Holly Taylor from Johns Hopkins University and its School of Public Health and the Berman Institute of Bioethics, and Aminu Yakubu from the Nigerian National Health Research Ethics Committee and the Health Ministry in Nigeria. We all know the basic framework that brings this issue before us. Since last spring, when the first cases began to appear, the Ebola outbreak took on a shape that is different than past Ebola outbreaks. Obviously, Guinea, Sierra Leone, and Liberia, the main affected countries, with some cases occurring elsewhere in Mali and Nigeria. These are the data that indicate where things stand according to the CDC as of Thursday of this week. A total of 17,256 cases, of which 10,000 plus have been confirmed by laboratory testing, slightly more than 6,100 deaths. There are cases that have also occurred in a number of other countries. As you know, in the United States, the CDC reports we've had 4 cases that occurred here. More have been treated here. We've had 1 death. In Mali, 8 cases and 6 deaths. There are 3 countries which WHO has declared had Ebola cases and are now free of them: Nigeria, Senegal, and Spain. And out of those 22 cases in total, there were 8 deaths. So we see a variable pattern from countries like Spain and the United States and Senegal, where the few cases that have occurred have mostly been successfully treated, and other countries where the death rate has been very high. I'm going to begin by having us look at the treatment side, and then we will look at issues of experimental vaccines. And I'd like to ask Holly Taylor to give us her view on the question of how experimental treatments should be used in this area of the outbreak with widespread transmission.
Good morning, everyone. It's great to see so many of you out there this early in the morning. And I have the pleasure and task to share with you some thoughts on, I'm really going to focus on the ethics in terms of thinking about conducting trials in the context of treatments for Ebola. And I thought I should sort of just give you a little context to say that I grew up professionally in the early and mid years of the HIV epidemic, and so my comments are certainly influenced by that experience. In full disclosure, I spent 2 years in Anthony Fauci's office as well, so that may be certainly an influence on my own development and thinking about these issues. But as I said, I'm going to focus on the ethical and leave some of the more complex science issues to others on the panel. I also thought it would be important for me to state up front that I think that it was unethical to distribute ZMapp in the way that it was. I think that it was inappropriate to give a compound that had not yet been tested in humans to humans who were desperate and had a fatal illness. That most of them, those who came to the United States, did not die, says little, I think, or nothing about the compound and whether or not it was a key to the survival of those individuals. In terms of thinking about the way generally we think about randomized controlled trials, I think we've made some progress. And again, part of the challenge here is talking about the US. This is very domestic in its focus. When I say we, I mean domestically, thinking about the challenges of people who are in desperate straits in terms of illness and our obligation to protect them, but also allowing them early access during the HIV epidemic. A number of parallel tracks were created in order to meet some of that need and our intuition to provide people in desperate straits with early access to promising therapies. I don't necessarily see a reason to abandon that general structure, nor the general structure that we use in terms of randomized controlled trials. So getting specifically to whether or not we should use randomized controlled trials for treatment for Ebola, I'll just mention as a point of fact that there have been in the press a number of compounds that have been considered or are being considered for in early phase clinical trials for the treatment of Ebola. There's survivor plasma, the idea of using the blood from those who have defeated the virus and hoping that might help others, and 2 antivirals that I'm probably not going to pronounce correctly, brindacidovir and favipiravir, both antivirals, that the idea being that it's going to interrupt the mechanism of the Ebola infection process. So again, thinking about the randomized controlled trial, right, the reason we do this, the reason why we develop controlled comparative trials is to compare a new thing to something that we have on hand in order to find out whether the new thing is better than what we have on hand. And the thing we have on hand these days is the background treatment that is, evolving as we speak in terms of what we provide those who have Ebola here in the United States when they come, and or the way in which we're developing that new standard in the context of the epidemic itself. And from my perspective, the most ethical and appropriate way to determine whether or not any new compound is the same or better than that background standard of care is to test that in a comparative controlled way. That it's, we all know that the randomized controlled trial is set out as what we refer to as the gold standard, and part of that is because it's the most efficient and effective way to get solid evidence. And this is an ethical trade-off that we established many years ago and continue to have in place and promote. That we, again, I mean sort of us in the room of those of us in the United States, agree to put a few patients in a trial, all of whom take on the benefits and burdens of that enrollment, and with the idea that their sacrifice and participation will benefit future patients. That is, if it turns out that the compound is better than the standard, future patient benefits. If it turns out that the new thing is not as good as the standard, that is, they do worse, that means we prevent future harm by not exposing future patients to that compound. And here I'll mention that, again, in the literature, in the sort of the media literature, there's been discussion of what I'll call adaptive models, in terms of clinical trials. And I think that that's certainly worthy of consideration, but I also think that we have to be careful about proposing something and maybe not providing the resources to facilitate the actual design and implementation of such a trial. That is, I'd want to make sure that we think about the trade-offs we might be making in terms of evidence. The other thing I'll say is that it turns out if you do the math generally, when we do a randomized controlled trial in terms of efficiency, we actually get evidence earlier than we might when we do a head-to-head comparison of two compounds without that controlled trial. So again, we're making some trade-offs in terms of sacrifice and burden. I'll, conclude this little part of my remarks just to say that I don't think there's an ethical justification to expose humans to an inferior design because of fears we might have about the way in which that might be perceived. And I'll transition to just say a couple of things about the humans that we are talking about, right? I am a advantaged, privileged white woman in the United States, and I certainly can't speak on behalf or well for those who are actually suffering from Ebola overseas, but I also think that we can get in trouble by saying things that also have been in the literature, that the people who live in those contexts might be unwilling or afraid to enroll in a randomized controlled trial that includes what I'll call the standard treatment. I think that there certainly are challenges regarding research literacy and understanding, but I don't think that's a reason to abandon our faith in the individual's ability to understand, and more importantly, our obligation to facilitate understanding. There's been a lot of discussion about trust, and I think while it's important, always important, to think about trust, I think it's important to think about the trade-offs of acting in a way that some might see as an inferior standard from the perspective of the Western viewpoint, and that doing that kind of trial can compromise trust in the same way that doing that trial might. So again, it's about trade-offs and thinking carefully about those. Thank you.
Good morning, all. And thank you very much for coming out so early in the morning. A demonstration of your commitment really to improve humanity. I want to start by saying that what I'm about to say, or the position which I am speaking from, is not that which says we are anti-RCT, if you would like. We agree that randomized controlled trial, either single blinded or double blinded, is really the gold standard. But what we are looking at it from is from the perspective that ethics, or bioethics if you would like, is one of the most beautiful things, in my opinion, that has happened to the field of science. What it did, or what it's expected to do, or what it is doing, is really to begin to look at or question or make the scientists, these geniuses that really come up with developments to improve health, to take a step backward, to look at the social context within which new innovations are to be provided, and to begin to think about the most appropriate ways of doing these things. And if you would like, Tony Fauci yesterday did mention how he was able to be converted into considering different approaches for doing clinical trials in HIV/AIDS. And these are some of the things that we think ethics or ethical discourse, or making people to think about differently from what they thought or what they think or what they hold staunchly that this is really what we need to do, to take a step backward to look at it. So I'll move forward from there. Yesterday in our presentation, our colleague from the Congo shared with us some of the cultural issues that are really the people hold. So he said that what people in the Congo consider interventions or healthcare workers is they call them white ghost bloodsuckers. And then we have another circumstance when we consider instances where a parent takes a child to a healthcare facility in a taxi which he couldn't pay for, and he had to wait in the car to see that child die because they were waiting on the queue. They were in bed spaces for them to get in. And we also consider the case of Mr. Abdul, if I'm not mistaken, who, after all the siblings had died and he had one daughter left, when he saw her being taken to the healthcare facility, perhaps he didn't know when he opened the door of the taxi and pulled her out and ran back into the jungle, they said. For me, these are descriptions about what people think about the healthcare system. What people, the fears about the healthcare system, they see going to the healthcare facility as a death sentence. So these people have lost hope completely in the system to be able to cater for their needs. So then we come to the issue of there's something out there that people are thinking might be able to help you, and that if you do take it, perhaps the death we're seeing in the healthcare facilities might reduce. Perhaps if your child or your wife goes to the healthcare facility, they wouldn't die because there's this new thing. So it's an issue about hope, it's an issue about building more trust in the system. Now, the ethical debates or the ethical arguments, from my perspective there too, Holly has rightfully identified that there's a need to do an RCT where you have people receiving an active treatment and the others receiving the gold standard. The gold standard today, as it were, we can see from the slides initially, so it's associated with nearly 60% fatality, although we are still improving on that. But what we even haven't done yet is really an analysis of what are the best strategies of delivering that gold standard, or what are the components of that gold standard that improve survival rates. So when we have something that holds some hope for the people, as I described earlier on, and then it is compared against a gold standard that for them, it really hasn't shown that much benefit. It is arguable that the principle of equipoise has not been fulfilled, and that has been, if you would like, one of the key things you consider when you're deciding whether you need to do an RCT or not. Now, the other argument is that of distributive justice. In our paper with some of our colleagues in Nigeria, we had argued that still within this context, that it will then really be unethical for you to decide you're going to give this treatment to some other people and not to others, especially when you have a community where families in chunks are really dying of Ebola, if you would like. And then there's this unhealthy situation in those communities that really facilitates the transmission of the disease from one community or from one family to the other, and so forth. But then the other consideration is that of the social economy or the practicality of really doing these things. Again, when we look at the issue of lack of trust, it is perhaps the case or a potential that when you decide you're going to isolate or you're going to really take out some people to give the treatments to, it's really possibly that that's going to increase that distrust or mistrust in the government, and perhaps also increase the rates of, if you would like, protests and breakdown of law and order. We have seen instances where people break into healthcare facilities to steal some of the medications from there in some other countries. Would it be likely that if then, in fact, they hear of this thing that is expected to help them better, that some of these things might increase? So these are some of the really positions that we are saying no, especially when these people have no hope and there's something that is giving them hope. We as scientists need to take a step backward to look at the delivery method. We're not anti-RCT, but we as scientists need to take a step back and look at the delivery method and look at how best to really address these issues. Thank you.
I'd like to invite Ed Cox to give a third perspective on the same topic.
Good morning, everybody. So this is a topic that we at FDA have thought about a fair bit over the last several months, and I'll try and just provide some perspective on the issue of studying therapies for the treatment of patients with Ebola virus disease. You know, as you've heard where we are today, I mean, we've got the largest outbreak of Ebola virus disease, and we really, at this point, don't yet have a therapy that's been shown to be safe and effective. The candidate molecules that are out there are in very early stages of development. Some are also in very limited supply, and in some cases, those supplies are intermittently available. We think about our goals here. What is it that we're trying to accomplish? Well, we're trying to help patients with Ebola virus disease. We're trying to figure out which among the interventions that one might choose can actually help people. It's also important to weed out the ones that will hurt folks and to try and do so as rapidly as possible in order to provide benefit to patients with EVD. And if we think about the most reliable and efficient way to understand what medical interventions are doing when administered to patients, I think it's well known that randomized controlled trials really are the most reliable way to definitively figure out what's going on with a particular medical intervention. There's also other things that come along with a trial, and that is that we would expect within a trial patients would also get good medical care. And that I'll come back to in just a minute, because that raises one of the challenges that would be encountered in designs other than a trial with a concurrent control. We've seen, if we look at patients who have come back here to the United States or who've gotten more advanced medical care, such as administration of IV fluids and electrolytes and blood products, what appears to be improved survival. Those that are in country taking care of patients have also, too, published on this and expect that better supportive care can improve outcomes. And it's important not to forget, too, that the therapies that are out there at this point have really not been shown to be safe and effective. And I'll share with you a story, and that is that as folks at NIH, NIAID were preparing for their clinical trial and are working towards that, in meeting with investigators, the opinions on the available investigational therapies that are out there and how docs that were using these therapies perceived their efficacy, their safety, was really remarkably different as you moved across the number of different investigators who'd used these therapies here in the United States. So let's talk a minute just about alternative trial designs. In the literature, there's a lot of discussion out there about randomized controlled trials, externally or historically controlled trials. But if we think about historically controlled trials, one of the real challenges here is that if you look at how a patient population is doing, you look at their mortality rate, then you institute a trial where you're going to bring in a medical intervention, but you're not going to have a control arm. And one case that you might see here is the 50% mortality rate that you see in the historical control may drop down to 20% once the trial's in place. In the setting of the trial, patients will be getting a particular medical intervention. In this case, it would be an investigational therapy. If that change goes from 50 to 20, what you won't know is that because patients got better IV fluids, better management of their electrolytes, the patients that were selected for the trial were not as sick, or their outcome, even without the medical therapy, would be much better. And that's what makes it really difficult to understand the results of an externally controlled trial. One of the other difficulties, too, if you continue to play this scenario out, is that once you have that result in hand, a 30% improvement in mortality, not knowing whether it's from improvement in supportive care or the investigational therapy, many will attribute that to the investigational therapy. And then you can play that out, which means that therapy then would be adopted, even if ineffective. You might subsequently do non-inferiority trials to an ineffective therapy. Other therapies that could have been effective may no longer be developed. So you can really understand the importance of getting this right. And now is the time to get it right, because it really is important that we establish the foundation for medical therapies here for Ebola virus disease and have that foundation to build on. Another thing with externally controlled trials, if you're going to do an externally controlled trial, you want to be able to have a comparable patient population. That is not always achievable, even if you collect a lot of data about patient characteristics. In this particular setting, where patients are being cared for in an Ebola treatment unit, it may be particularly difficult to collect all of that information that one might want to collect. In a randomized controlled trial, randomization is so powerful in helping you to get to comparable patient groups, there can be tremendous economy in the amount of data that you need to collect about patient populations. With regards to RCTs too, very important that some of the advances that are out there are incorporated into the RCT design so that once something is shown to be effective, we can recognize it as soon as possible. Bayesian designs, monitoring of the trial. The NIH trial talks about looking after sequential patients are included to see if a boundary is crossed somewhere along the way so that the trial is really stopped once efficacy has been shown. Once an agent is shown to be effective, it could be incorporated into the standard of care, provided there's adequate supply. Another question I'll just touch on really quickly is people, have asked, why do the trial in the U.S.? Why not just do it in West Africa? And at least as I think about it, I think it's important to be able to, do the same type of trial here in the U.S. that we would also pursue within West Africa. So in essence, that all are treated equally. I've already talked about how important it is to get this right. And one other thing too, with the very limited supplies that are out there, there's discussion in the literature too about, it would be difficult for only some patients to get, investigational therapy and others to not. Well, that's really a supply issue because even if you do an externally controlled trial, the number of patients with the disease will still greatly exceed the available supply of medication. So in a randomized controlled trial, in essence, it's a lottery, and that's how the products would become available, would be through random allocation. You provide a fair means to make the product available. If you only have 50, 60, 70 treatment courses that you can utilize, if those treatment courses are utilized in a controlled trial where you can learn, you can actually benefit many people. If those same number of treatment courses are distributed through an externally controlled trial or even in the absence of an externally controlled trial, it'll be hard to learn nearly as much as you could with a randomized controlled trial. You might potentially mislead yourself. So I just want to bring to bear the point that the limited supply also brings additional considerations to the fore. So not an easy area. It's going to take real leadership to be able to do such trials. Important that we engage the health authorities in the countries and also the communities involved. And I think this is really our best strategy to be able to help patients. And I also, too, just want to applaud our colleagues at the National Institutes of Health, NIAID, for their work and leadership in the area of trying to get randomized controlled trials in place. Thanks for your attention.
Now, before we move on to talk about vaccines, I want to think about this subject from what seems to me is the viewpoint of an IRB. I want to hypothesize that there are two groups of investigators in the United States connected to your institution. One group is planning to cooperate and be part of the trial that the NIH is planning to do here in this country and then possibly in Liberia if arrangements can be made. The other group is associated with MSF and WHO and the Wellcome Trust that is planning to do treatment in Guinea and Sierra Leone. What do? We've heard from, I think, both from Holly, putting it in ethical terms, and from Ed, putting it in terms of the scientific payoff and both the speed with which you'll have reliable information and the quality of that information. We've heard the sense that an RCT is important. And I want to ask you both whether we would be really, in effect, talking about a placebo control in the following way. If in the centers where this is going to be done, we arrive in those centers, at the highest possible, in that setting, level of basic supportive care, so that all the patients, whether or not they get the intervention, will get the best possible care that can be delivered there. Would it be acceptable to have a design in which one arm got that and the other arm got that plus whichever one of those drugs or the convalescent serum that you described?
So is this on?
Yes, it is.
So, yes, that's exactly what I'm referring to in support of sort of traditional randomized control trial, that the expectation would be that the standard arm receive whatever the investigators in consultation with all the folks who are treating Ebola in country and have treated Ebola here in the United States, that those randomized to that arm would get the best care available in that setting, with the assumption is that it's the best care that one would get. And that everybody gets that, then half the folks are randomized to get whatever we think is the new thing versus that.
Ed, similar answer or different answer?
Yeah, similar answer. Best available supportive care, both arms of the trial, the investigational drug in one of the two arms by random allocation, and best available supportive care from what we know should provide benefit to folks too. And the real question that this type of trial would address, an appropriate question is the investigational therapy adding value to what's already being done?
Now, if we had not had in the United States what amounts to basically 100%, I mean, that one case, I gather, was one that got into the treatment very late. But if we had not had sort of that dramatic sense that, in fact, you can do a lot with that, the position you took, Amino, was based, as I understand it, on the notion that the basic treatment available in these countries has a 60 or 70% death rate. And you can't talk about that being in equipoise with any intervention that people would be confident enough in that they would be trying to test it. Is that your position?
Yes.
It's the absence of equipoise here.
Yes.
Okay.
So two things,
But
actually.
let me ask then Holly and Ed another question, which is the practical difficulties that Amino brought up, the notion that you will lose people coming in for treatment because they will say, if you're giving some of us a drug that you're testing because it's supposed to help and others aren't getting it, by your choice, not because it's not available, but by your choice, you're sorting people out, you will have a lack of trust. And that lack of trust back in the early AIDS era led to people pooling their medicines and saying, we will not accept this randomization. We will not accept trials. We are in a crisis. How is this any different?
Yeah, so two things. One is I think that it's important to think about how we use the term equipoise. I don't think that we can almost generally say some medical intervention is better than none. But when you have a fatal illness like Ebola, like HIV, like a life-threatening cancer, et cetera, it's not the case, I don't think, that we have equipoise that there are no good treatments. I don't know that we have enough evidence on at least the two antivirals that we have in hand. I don't think there's much agreement that these will work. They might work, but that doesn't mean they will work. I completely appreciate and understand the practical challenges in conducting the research in the setting that we're talking about and the low resources available. That said, I don't anticipate that these trials are going to be done, or maybe I'll say it another way. My anticipation is that these trials will be done in a way that makes sure that those who are in the trial have the best available care that they might receive here in the United States comparatively while on that trial. That we're not talking about saying, let's give them, what we have on hand and see what happens. I think to say that the infrastructure may be compromised, I think it would be unethical to build the capacity there to not do the best trial that we could. I also don't think that it's necessarily true that the fact that there is mistrust means that we have to compromise our approach in terms of conducting the trial. I just can't imagine, I mean, again, and this may just be my lack of imagination, I can't imagine that on one hand, you can say that individuals are demanding access to treatment, and on the other hand say no one will consider enrollment in that clinical trial. You're absolutely correct that in the early days of the HIV epidemic, folks pooled their drugs. That said, we were able to successfully complete randomized controlled trials to find out that AZT and then AZT as the standard arm of many future trials were conducted and found to be helpful. So I think that there's absolute parallels, and the parallel story is we were able to accomplish it, and despite, the challenges along the way.
I wanted to look for an exchange between Amino and Ed on this issue. Amino made such a strong case about the basic injustice in a situation where you're picking out people, and Ed's response was, the benefit to people is that knowledge that you get, that learning is the benefit. That's what benefits people in the long run, having reliable information. That's in a way saying to patients who would become subjects in trials, you're in this trial so that somebody later, it's the classic, someone later will benefit. Is there any sense here that this epidemic, with better supportive care, will come under control without any new drugs? And if that's the case, are people who are developing the drugs in effect saying, we'll lose this opportunity? So they're really saying, we want to get in there while we can to develop knowledge about how to treat Ebola for the future. And we're almost afraid that the epidemic will burn itself out before we get that knowledge. That puts a different cast on what we're asking people to do, it seems to me. Ed, do you have any sense? Because you were emphasizing this, the knowledge side. Learning benefits people, you said.
Right, and I think that's part of it. If we think about agents, many of the agents that folks are looking at are in limited supply. So, one of the fairest way to make those agents available is either through a lottery or random allocation. So there is a certain degree of fairness, in essence, inherently in the design of the way drugs would be allocated in a randomized trial. In addition, if it's not an appropriately designed trial, the trial may not have the capacity to detect harm. So that harm may go on, and if those drugs had been used in an appropriately designed trial, you might be able to stop the trial, monitor the trial, do something to prevent harm from going on. If that trial completes and the harm still isn't detected, then that harm gets transferred to populations. Yes, the knowledge gained from the trial will benefit future patients, no question, from a properly designed trial. It may also help patients who would have been part of a trial that stopped early. As far as the epidemic, I think we all hope that this epidemic ends as soon as possible, no question about that. And infection control measures will be critically important. We may learn something about therapies, but I do think that traditional infection control procedures, contact tracing, monitoring will be absolutely key in trying to bring this epidemic under control as soon as possible.
And I want to get, before I go to Walter, I want to get a response from Amino. As Ed says, these drugs are likely to be in short supply. And so if you're having to use a lottery or some other even-handed way of distributing them, why not do that as part of a trial? Why couldn't people be convinced by the governments, we're doing it this way because we want to be fair, and so it's drawing names out of a hat, as it were. It's a random allocation is the fairest way of doing it.
Thank you, Alex. I think one of the key issues is the urgency with which we want to do this. So if we really want to get people to buy into these things, we need to first begin with building trust. And it takes a lot of, it will take a while to be able to build that trust, to be able to convince people. So what we're saying is, we're also not saying automatically don't consider these, but in the interim, while the disease is still raging, when it is very difficult for you to really build that trust today or tomorrow or next tomorrow, that perhaps what we could start doing is, and again, this is another issue, that what is the optimal combination of IV and electrolyte replacement that optimizes the patient outcomes? We don't know about that yet. And what we're saying is perhaps we should begin with that, so we have a baseline that says, okay, this is the baseline mortality when you provide that optimal.
But would you randomize the assignment to different therapies of that sort, the different supportive therapies?
So the randomization will come after we've been able to establish a baseline. So that's a standard combination of electrolyte and IV replacement, and other supportive care if you would like. And then after we had also, if you would like, on compassionate grounds, been able to provide some of these limited drugs, treatments to some people, and be able to compare the outcomes between one or the other. So quickly we have an idea of which one will work, which wouldn't work. And those then, together with the observational data, we could then use to put into a trial, an adaptive trial design.
Now, we heard yesterday from Dr. Fauci comments about vaccines and the special characteristics, which we all know, that in vaccines, you're giving a substance to healthy people to prevent something, you hope, but as he illustrated, sometimes very promising vaccines turn out not to work. And as we put this panel together, we have the benefit of having Walter Strauss, who has global responsibility for Merck's vaccine work. And then we got the complication that Merck now has one of the vaccines, the two vaccines that are being looked at in its portfolio. So we have to give Walter the blessing of no conflicts of interest here, because I think he can declare whatever conflicts he feels. But when we put him on the panel, there was no conflict, and then the company created one. Walter, I assume that your view vis-a-vis vaccines is the same as what Ed and Holly have said about the need for a randomized controlled trial. But are there any other aspects of this picture that you would like to emphasize in considering experimental vaccines?
Yeah, thanks, Alex. So, after we came up with this clever idea to have this morning session, Merck entered into an agreement for a experimental Ebola vaccine. I'm going to offer you my personal opinions. These are not necessarily the opinion of the company, and I'm not going to be discussing the particular experimental vaccine. There are a couple of key points that I think that one needs to consider in looking at vaccines versus therapeutics. The first is that we're talking about prevention as opposed to treatment, okay? The second is that, generally speaking, vaccine trials they tend to be larger, but in some ways they're conceptually easier than our drug trials, in that typically 1 to maybe 3 doses are required. The outcomes are generally pretty straightforward, and any sort of safety issues that generally occur shortly after administration of a vaccine, and one sees different scenarios sometimes on the drug side. I do very firmly believe that controlled trials are going to be necessary in order to ascertain whether an Ebola vaccine is safe and efficacious, and I very much agree with the remarks that Ed made around adaptive trial designs. I think there are opportunities to have more efficient trials than customary, but the basic principles are still valid. So I wanted to put this into a context. I'd like to ask you to recall Dr. Fauci's remarks yesterday. And his remarks are around the STEP trial, which was considered to be the most promising HIV vaccine trial at the time, is one that I and Merck were involved in. And he mentioned it in his typical Dr. Fauci rapid-fire style. But I wanted to just sort of ask you I want to pause for a second and highlight a couple of points. The first is that this vaccine trial was based upon years of internal work, and it wasn't piecemeal work. This was one of the key priorities of the company that I work for. And it was a novel vaccine design which took an adenovirus vector, which was modified so it would be weakened, and then in which a couple of proteins from HIV, antigens from HIV, were attached. And it was considered, and so the adenovirus was considered to be essentially an entry mechanism for the vaccine, and then the HIV antigens would evoke a response. On the basis of extensive non-human primate data, as well as phase 1, very promising phase 1 human data, a set of very complex negotiations took place. The NIH was keenly interested in this. I can assure you that if there's one thing that Tony Fauci wants in his career, it is to have a successful HIV vaccine. The NIH threw its full support behind this. Leading scientists around the world in HIV entered into into planning this vaccine trial. And arguably, HIV is one of the most, if not the most, studied viruses in the world. So there was a tremendous, tremendous amount of brain power that was applied to designing this trial. And then what happened? Well, Tony mentioned that the trial was stopped on account of futility, and not only that, but the individuals who had received it was a placebo-controlled trial, the individuals who received the experimental vaccine actually had a worse outcome. They had a higher incidence of HIV than did the control arm. What a surprise. And what we learned as a result of this, we're still learning, but basically we learned that this model, the adenovirus, was not the kind of innocent bystander that we thought it was. So the key point is if one had taken some of the arguments that are being made in the press today and simply saying, well, we have promising data from early research, let's just go out and distribute this vaccine on some sort of compassionate use basis, and God forbid the vaccine was not as safe as we hope it to be, we might wind up with a worse outcome. Not necessarily Ebola, it might be some other safety issue. And in countries where the infrastructure is as fragmented as it is in these Ebola endemic areas, we might not have the ability to know that for quite a while. So those are kind of key points. And then I'd like to just close by saying that in contrast to HIV, which was so heavily studied, we know pretty little about Ebola. And one of the reasons for that is it is such a so-called hot virus that it has to be studied in the lab under very, very controlled circumstances. And there are only a few labs in the country which are authorized to conduct this sort of research. We also don't know much about the natural history of Ebola because the outbreaks that have occurred in the past have occurred in relatively remote areas where the virulence of the virus has caused devastation in a couple of villages, but then it's gone away. What's different about this particular outbreak is that it's traveled to several different countries. It's, unfortunately, I guess, afforded us the opportunity to learn much more about what can be done for Ebola, unfortunately in the sense that there are many more cases. But we've learned that the classical wisdom that 60 to 70% of people who get infected with Ebola die, that if you intervene early with hydration and electrolyte therapy, as Amino has alluded to, you can drive that case fatality rate down to 25%. And that's just on the basis of what we've learned from these active interventions in emergency settings. So the standard of care, if you will, is evolving. And that's one of the real challenges of doing an uncontrolled trial or doing a trial where you're referencing historical controls, because your standard of care is continuing to change. So I'll pause there.
Thank you. Now, Amino, are you thinking of vaccines being tried out in a distribution model or a randomized controlled trial?
So our response to this, so the first is we had considered, first of all, really the target population that has been initially proposed. And our position then was, if you have healthcare workers, we're saying that they're at-risk population, that they probably may be able to understand when you say this is something that will potentially prevent and things like that, that it's easier to enroll them. But our initial argument is that you shouldn't restrict that to just healthcare workers, because the people living in these slums and the families, they're really more at risk of contracting the disease. To get results in a vaccine clinical trial, you need the potential of seroconversions. And it is perhaps best when you include people within the communities and families that, within the communities most affected with the disease than just restricting it to healthcare workers. On the case of whether it's a randomized controlled trial or distribution, in my own discussion with some of my colleagues, we had preferred, if you would like, an adaptive design, where, for example, you have one community that you randomize and give people, but you use a different community with similar kind of demographics and prevalence as a control community. And it's for the same concerns that if the same community gets to know that you're giving some people something, you're not giving some others something, it's still the same concerns I raised with the treatment might hold. So one of the easiest ways we thought of doing that is having another community, perhaps even in another different country, one of the Ebola endemic countries, as a control that you would use. Thank you.
So Holly, from your point of view, is that sort of cluster randomization, is that acceptable? Just yes or no?
Maybe.
Maybe. And Ed, from the FDA's point of view, cluster randomization or too many unknown differences from one spot to another?
Right. So if you start to think about it, there's pros and cons with every trial. It gets, it does provide a control group. One of the questions will always be, what was the rate of transmission within one population compared to another? And if there are differences, that can certainly make it a little more challenging. As you get more and more sites, maybe that will help. But, with any trial design, there always are pros and cons.
So in your IRBs, you'll have 20 minutes to deal with this protocol, as you do with all the other complicated protocols. You'll have these scientific issues about what you can learn and can't learn from one design or another, what is politically, socially, and ethically acceptable in communities. I hope that these exchanges, all of them very respectful and calm, but very different in their approaches, have helped to shed light not only on this, but there will be other problems, unfortunately, I'm sure we'll be faced with, where things have to be done quickly. We don't have the knowledge of the disease, which is evolving itself, the treatment is evolving, and investigators are going to have to come forward and convince you that whichever approach they're going to take is ethically acceptable. I want to thank our panel for coming in early in the morning.